The purpose of this presentation is to present the AROCELL TK 210 ELISA test, which is a unique biomarker for tumor cell proliferation. The rate of tumor cell proliferation is an important biological parameter that may aid in cancer management and may show how fast the tumor is growing or not. We say that we measure what really matters. During the course of this presentation, we will review the product background in research and development, the potential of TK210 ELISA in determining serum TK1 protein, the potential of TK210 ELISA compared to TK1 activity tests and other biomarkers, such as CA15-3 and Ki67, sample storage, a brief description of AROCELL as manufacturer, and finally, how to obtain more detailed information about the AROCELL TK210 ELISA test. Professor Staffan Eriksson is a co-founder of AROCELL and one of the researchers and inventors involved in the development of the TK210 ELISA test. He is a pioneer in the field of the structure and function of thymidine kinase, with over 120 studies on TK1 as it is commonly called. He has been involved from the very beginning with the first scientific studies defining the molecular structure of human TK1 in 2004. He has fundamentally contributed to the current knowledge of TK1 structure and function related to cell proliferation. In recent years, he has focused on TK1 as a biomarker in different tumor diseases using antibody-based methods. He is inventor of 3 of AROCELL's international patent and patent applications in this field. For over 3 decades, there has been research conducted on TK1 in many different types of tumors. TK1 serum activity is a valuable biomarker. However, enzymatic activity assays have limitations relating to specificity and sensitivity, as serums contain inhibitors of TK1 activity, and the nature of TK1 to exist in multiple forms with different specific activities. An immunoassay for measuring TK1 has been envisioned for a long time in order to solve the problems of TK1 enzymatic activity assays. TK1 immunoassays simplify sample handling, improve sample stability, and are more robust assays in the routine laboratory environment. This provides a new potential for application in routine laboratory settings in standard clinical use. This vision inspired the researchers and co-founders of AROCELL to develop and deliver a better TK1 immunoassay based on 2 monoclonal antibodies specific for a unique TK1 amino acid sequence, the TK210. Development was started in 2003 and resulted in the first ELISA prototype in 2008. The finished product, the AROCELL TK210 ELISA test, was CE marked in 2015 and is commercially available within the European Union. TK210 ELISA is available in the U.S. for investigational purposes only. In order to explain the potential of TK210 ELISA and how it works, we must illustrate the disease process on a cellular level. TK1 is a key enzyme in the synthesis of DNA, and it is upregulated during tumor cell growth. This image illustrates a cross-section of a solid tumor. Cells multiply within the growing tumor. Cells also die during the process, and that is how TK1 is released during cell turnover. In the blood, TK1 occurs in the form of complexes of multiple units of TK1 with a wide range of molecular weights and enzyme activities. Now let's leave the cellular level. This animation illustrates how thymidine kinase is released within the body from a breast tumor into the bloodstream. This is why it is possible to measure TK1 in an easily accessible serum sample to see if the tumor is growing or not. Information about cell proliferation is very valuable, and in combination with a cancer diagnosis, it may aid in prognosis and therapy selection, and is why we say measure what really matters. The TK210 ELISA test is unique as it measures TK1 protein concentration in serum. To give a short orientation, in the category of biomarkers measuring cell proliferation, we have the traditional TK1 activity test that we mentioned earlier. It is a serum biomarker like the TK210 ELISA and has been used for many years mainly in research. We also have the gold standard Ki-67 biomarker, used routinely in tumor prognosis assessment. Ki-67 is a tissue biomarker, which means a tissue biopsy is needed from the tumor. We know that TK1, when used as a tissue biomarker, correlates highly with Ki-67. The issue with tissue biomarkers is that the tumor can be big and heterogeneous, and only a small part will be covered in the testing procedure, which means that the highly proliferative parts of the tumor can be missed. This is the same with the needle biopsy. It is only representative of a very small part of the tumor. A serum biomarker represents tumors present in the entire body. Among the biomarkers that measure tumor mass, we find most of the tumor-specific biomarkers like CA15-3, PSA, and others. Monitoring the tumor size is important, but knowing the tumor cell proliferation rate may be equally important. Biomarkers often complement each other, and there is a trend to use biomarker panels for cancer management. TK1 has been studied in many different types of tumors, as illustrated by this table. Several studies have included the relationship between TK1 and the stage and grade of tumor disease. TK210 ELISA is a very broad biomarker and will most probably be used as a complementary biomarker together with other tumor-specific biomarkers, as you will see in the following slides. Here we are proud to present the first publication of the CE-marked TK210 ELISA test. It was published in the journal Tumor Biology 2016. TK1 was studied in sera from women with breast cancer, and a comparison was made to the specific breast cancer tumor biomarker CA15-3. The TK210 ELISA levels were measured in relation to different stages of breast cancer. The analysis shows that the test is sensitive and specific. This means that TK210 ELISA was able to identify raised levels of TK1 in different stages of breast cancer with statistical significance. Keep in mind that you want to detect levels in the early stages of cancer progression. Here is the comparison between TK210 ELISA and the breast cancer-specific biomarker CA15-3. It is illustrated by a ROC curve, where the sensitivity is plotted against the specificity. The curve shows how well a test performs, if it is sensitive without picking up false positives. A high area under the curve, or AUC values, indicates a good performance. TK210 ELISA, CA15-3, and their combination were measured in women with stage T2 breast cancer. Interestingly, TK210 ELISA showed greater area under the curve than the classic tumor biomarker CA15-3 alone. However, the real benefit is showing when combining TK210 ELISA and CA15-3. The combination increased the area under the curve to 0.94. The take-home message from this analysis is that the highest prognostic power is found in the combination of the two biomarkers, measuring tumor cell proliferation and tumor mass. This shows the potential value of adding TK210 ELISA to existing biomarker panels. When studying tumor biomarkers, sample stability is very important in that it simplifies handling and shipping, and in the case of research, it can speed validation by enabling the study of archived samples. This table shows that the serum TK1 immunoreactivity is very stable when measured with the AROCELL TK210 ELISA. Samples can be left in room temperature for 24 hours or kept in the refrigerator for 5 days. This makes the AROCELL TK210 ELISA highly suitable for clinical trials. The AROCELL TK210 ELISA test is the only CE-marked ELISA for thymidine kinase 1. CE marking for the test was obtained in 2015. The product is protected by 12 patents, and the most recent patent application protects the sequence of 3 monoclonal antibodies for use in the immunoassay for TK1. TK1 is an early biomarker of proliferation. Serum levels may reflect cellular proliferation. TK1 is a broad marker. It can be used to study hematological malignancies and various tumor types. Routine ELISA procedure means that standard clinical chemistry labs need no special equipment or new methods. It is easy to automate. Better discrimination between healthy controls and subjects with solid tumors. Positive synergy with existing tumor markers. TK1 may aid in prognosis and therapy selection. TK210 ELISA is developed and manufactured by the company AROCELL AB, located in Uppsala, Sweden. The company was originally a spin-off from the Swedish University of Agricultural Sciences and the Karolinska Institute. AROCELL AB is listed on the Nasdaq Nordic Stock Exchange. For more in-depth information about the TK210 ELISA test, please refer to the AROCELL.com webpage or welcome to contact us at info@arocell.com. Thank you for listening.